Welcome to CJ Bio's YouTube channel. I'm Jason Lee with CJ Bio America, and I'm going to talk about the importance of tryptophan in laying hen and broiler breeder diets. Tryptophan is an essential amino acid that is involved in protein synthesis and plays a key physiological role in many molecules, including melatonin and serotonin. Tryptophan regulates insulin-like growth factor 1, corticosterone, cortisol, and regulates the secretion of heat shock protein 70. Tryptophan and its metabolites regulate feed intake, reproduction, immunity, neurological function, and anti-stress responses. Tryptophan deficiencies could lead to decreases in growth performance, compromised immune status, and behavioral changes within the animal. All large neutral amino acids, such as tryptophan and branched-chain amino acids to include leucine, share the same brain transporters. Corn-based diets, which are used in the U.S. and many other geographical locations around the world, contain excess amounts of leucine due to the high content of leucine present in corn as a percentage of protein. Laying hens and broiler breeders that are fed corn-based diets can contain an excess of 180 digestible leucine to digestible lysine ratio, when the requirement is between 110 and 120. Therefore, excess leucine in these diets may decrease tryptophan absorption and impact feed intake and serotonin concentration. In a published study that evaluated the effect of large neutral amino acid concentration on the tryptophan requirement in laying hens, it was observed that a 40% increase in large neutral amino acid concentration increased the tryptophan requirement from 178 milligrams per day to 212 milligrams per day, which is a 20% increase in the tryptophan requirement. And this chart shows the performance of Ross broiler, breeder male and female parent stock, and the target weights that we strive for at different ages. The blue line represents the target body weight of the Ross broiler 708 straight run. As you can see, a target body weight of 8 pounds is achieved at 7 weeks of age. However, the Ross 708 male, parent stock male, and parent stock female has a similar body weight target of 8 pounds at 23 and 41 weeks of age. This large difference provides challenges for integrators in rearing replacement broiler, breeder, male, and female birds. Thus, body weight is closely monitored when rearing broiler breeder pullets. Excess fleshing, if developed, will negatively impact breeder behavior and flock fertility and reduce the number of chicks that are received. Thus, lysine intake in replacement breeders is closely monitored and needs to be considered during dietary formulation. The table at the right shows 2 different pullet grower diets using corn distillers, wheat midds, and soybean meal as our bulk ingredients and free amino acids to meet the amino acid requirements. We maximize our crude protein level at 15.5% with an AME target of 1,275 kcal per pound. We maximize our digestible lysine at 0.61% of the diet, and these restrictions on lysine and crude protein provide challenges in meeting the lower amino acid recommendations, such as digestible tryptophan. In our first diet, our digestible tryptophan solves at 0.12% digestible tryp, which is lower than the Avegen recommendation of 0.15% digestible tryp, which is achieved in the second diet. We achieved this by the addition of L-tryptophan into the pullet grower diet with 0.02% tryptophan. This represents a 20% difference between the digestible tryptophan levels of these 2 diets. I also want to point your attention to the digestible leucine value of these 2 pullet diets. Both are in excess of 200% digestible leucine to digestible lysine, indicating a large excess of digestible leucine present in these pullet grower diets. The question is, how could this excess leucine influence our digestible tryp requirement and concentration needed in our pullet grower diets? Feather pecking and other behaviors in laying hens, broiler breeders, or replacement breeder stock can impact performance and welfare. In a study in 2004, a group in Europe reported that the acute reduction of serotonin turnover in the forebrain increased gentle and severe feather pecking behavior, connecting the behavior of feather picking to serotonin concentration. Low serotonin content can be related to low dietary tryptophan or an antagonism with other large neutral amino acids at the blood-brain barrier, such as leucine. Thus, a potential solution would be to increase tryptophan intake, to increase plasma tryptophan, and increase tryptophan competition to access to the brain. A research group in Europe in 2019 published a paper evaluating the effect of tryptophan depletion and the influence of that on the social behavior of hens. They utilized 2 amino acid premixes, one with adequate tryptophan and one absent of tryptophan, and fed to 2 different populations of hens, and found that hens fed tryptophan depleted premixes had a higher incidences of pecks per minute, connecting the difference in presence of tryptophan to the behavior of the animal. Similarly, Shea and colleagues published a paper in 1990 evaluating the effect of adding L-tryptophan into diets of developing and mature broiler breeder males. They conducted 2 studies, the first with a control diet with the addition of 0.75% tryptophan and 1.5% tryptophan. And then in a second experiment, they added a 4th treatment group, 0.38% tryptophan. In both studies, it was observed that adding L-tryptophan decreased the threats per bird and the pecks per bird that were observed in these populations of mature broiler breeder males. Similarly, treatment, a 2% added L-tryptophan in the diet. The charts at the left indicate that, in fact, genetic differences were observed between the 2 genetic lines. And in the high feather propensity population, the addition of 2% added L-tryptophan significantly reduced the gentle feather pecking incidences within that population, and it reduced them by half. Chart at the right indicates the plasma tryptophan to large neutral amino acid ratio present. Thus, as tryptophan was added into the diet, an increase in the ratio of tryptophan to large neutral amino acids is increased, thus increasing the competition of tryptophan for access to the brain. Thus, multiple experiments have shown the benefit of added L-tryptophan on behavior and serotonin concentration within different populations of birds. However, the majority of these experiments have focused on concentrations of L-tryptophan which may not be commercially available or commercially viable. Thus, at CJ Bio, we work together with Texas A&M University and Dr. Greg Archer's lab to evaluate some lower levels of tryptophan inclusion, resulting in lower concentrations of digestible tryptophan in the diet and its results on animal bird behavior and serotonin concentration. Thus, in this experiment, we utilized 4-week-old raw 708 pullets for a 6-week evaluation period, 3 birds of each treatment with 13 birds per replicate pen with 5 different levels of tryptophan concentration, ranging from 0.11% to 0.50% digestible tryp, which were related to an inclusion of 0 to 8 pounds per ton inclusion of L-tryptophan. We evaluated behavioral assessment at 3 weeks of age and serotonin concentration at 6 weeks of age. Beginning with the serotonin concentration, we can see that adding L-tryptophan into the diet increased the serotonin concentration of all treatment groups. In assessing the behavioral assessment, we utilized a tonic immobility to test the fear assessment of these birds. For those of you unfamiliar with tonic immobility, you take a bird, you present it into a foreign environment, you enter the bird into tonic immobility by laying it upside down. To assess the influence of L-tryptophan on bird behavior, tonic immobility was utilized to determine the fear in these broiler breeder pullets. The latency to first head movement and the duration of time to movement indicates the level of fear a bird is expressed to. Thus, if we look at the control feed, we see a latency to first head movement of 110 seconds and a quadratic reduction in the amount of time to first head movement as we increase the amount of L-tryptophan in the diet. This indicates that the birds are less fearful as they are moving sooner. So this small research project led us to partner with some integrators to evaluate the use of L-tryptophan in the field. Obviously, there's a much different environment in field conditions as opposed to a controlled experimental setting. Thus, this allowed us to evaluate the strategy of adding L-tryptophan into pullet diets in a commercial setting. In this first field trial, We selected neighboring complexes from a large integrator, one complex on a traditional rearing program, and one complex on a traditional rearing program with the addition of L-trip in the feed. Both complexes are fed out of the same mill. Thus, the same ingredient profile is used. The only difference is the presence or absence of L-trip. We sampled these birds 2 different days, 24 hours apart, day 1 being an on-feed day and day 2 being an off-feed day. As you can see on day 1, the control bar is the blue bar, and the test diet, which is the inclusion of L-tryptophan, is the orange bar. Adding L-tryptophan to the diet increased serotonin concentration of the birds fed L-tryptophan. On day 2, which is 24 hours post feeding or post the day 1 sampling, we observed a large reduction in the serotonin concentration between day 1 and day 2. And we also observed a large increase in the amount of serotonin present in birds fed the L-tryptophan in their feed. Interesting, the decrease in serotonin concentration between day 1 and day 2 was much larger for the control group as compared to the test group. And if we look at the test group on an off-feed day, the level of serotonin is similar to that of the control birds on an on-feed day. We then had the opportunity to partner with another integrator in a paired house study, so kind of a go-between or between a full-fledged field trial utilizing an entire complex and an academic field trial or an academic research trial. We utilized pullets and males, which were placed in house 1 and 2, were fed a control feed, and house 3 and 4 were fed the experimental diet. We fed a common starter diet through 28 days, and the treatment feed started on 29 days and was fed through the point in time when the pullets were moved to the breeder house. At 12 weeks of age, which was the same week that we sampled the birds in the first field trial, we went and sampled the birds for serotonin concentration in this experiment as well. Serotonin concentration is listed on the x-axis here. On the skip day, we sampled skip day first, and the on-feed day, the second sampling day. Between the control and the treatment, we can see significant differences in serotonin concentration with the addition of L-tryptophan in the diet. On an on-feed day, similar separation or significant differences are observed with the addition of L-tryptophan compared to the control feed. Interestingly enough, on a skip day, the birds fed L-tryptophan actually had a higher serotonin concentration than compared to birds on the control feed on an on-feed day. And similar results were observed for the males in this experiment as well. Serotonin concentration on the control was higher in the on-feed day as compared to the off-feed day. However, adding L-trip into the diet, regardless if it was a skip-a-day or on-feed day, led to significant increases in serotonin concentration. Behavioral problems such as feather pecking are associated with low serotonin concentration. Low plasma trip levels or competition with other large neutral amino acids at the blood-brain barrier can reduce the serotonin concentration. Adding L-tryptophan to laying hen and broiler breeder diets increases serotonin concentration and influences the bird behavior. Regarding inclusion rate, can be variable and based on numerous conditions, including ingredient profile, management, conditions in the house, and behavior of the birds. However, targeting a digestible trip concentration of between 0.25 and 0.3% in breeders and 0.2% in laying hens will increase your serotonin levels. I'd like to thank you for your time. I'd also like to invite you to visit our YouTube channel, where you can see other interesting talks from experts within CJ globally.